My name's Sharland. I am a transplant researcher from the University of Sydney. My background is in nephrology, internal medicine and transplantation. And the reason I work in biomedical research is to try and improve outcomes for transplant recipients. There are a number of grand challenges in transplantation. One that I see is that while the short-term outcomes of transplantation are really excellent, the long-term outcomes are still somewhat disappointing. So we know that around 40% of kidney transplants are going to be lost within 10 years after transplantation, and for lung transplants the outcome is even worse. So 55% of those grafts will not be functioning within 5 years. Both the innate and the adaptive immune system are important for determining long-term graft outcomes, but our focus is principally on the adaptive immune system. I think despite the introduction of ever more powerful immunosuppressive agents, they really haven't been able to substantially improve the long-term outcomes of transplantation. And so in order to do that, we really need to understand the molecular pathogenesis of graft rejection. And that starts with understanding the ligands that are recognised by alloreactive T cells and B cells, and the repertoire of those immune cells. So the composition of the immune cells in the recipient that are causing graft rejection and how they change over time. We know that acute T cell mediated rejection affects up to 30% of kidney graft recipients, and even in its mildest form, it's associated with a worse long-term outcome than that for grafts that haven't undergone acute rejection. So I'm now part of a clinical trial called Tackle It, and the aim of Tackle It is to get the best possible information we can about the treatment of T-cell mediated graft rejection and whether or not the treatment results in resolution of rejection. As part of the study, I'll be looking at the T-cell receptor repertoire and seeing whether changes in the repertoire correlate with resolution or not of rejection and the outcomes of treatment. So in order to do this, we are partnering with Omniscope to determine paired alpha-beta T-cell receptor chains in the peripheral blood of our recipients matched with information from the renal biopsy and the infiltrate of the renal biopsy at scale.